the first talk will be by Stefan Kubick, who is in the field of cell-free synthesis of transmembrane proteins for quite some time now. He was group leader at the Fraunhofer Institute in Potsdam-Golm for quite some time, but now ventured on to found his own company. He's CEO and founder of the B4 company. It's B4 PharmaTech, and he will now give us an overview about what this company can do about cell-free systems for the production and functional characterization of membrane proteins. Stefan, please.
Yeah, Konrad, thank you very much for the kind introduction, and thank you very much for having me here, and thank you for showing up that early in the morning after such a beautiful dinner. The company is B4 PharmaTech, as Konrad already mentioned. The company is located here in the southwestern part of Berlin on the campus of the Freie Universität Berlin. So on the one hand side, I'm a lecturer at the university. On the other side, I'm the CEO of the company. On the left-hand side of my desk is the pure academic research. On the right-hand side of my desk is pure industry business. So the industry business is more or less the protein production service. So we also offer a service for the production of membrane proteins and difficult to express proteins, but also antibodies, fluorophores, and labels are integrated in the protein of interest. And we synthesize a huge array of different linker molecules that can be attached by click chemistry to membrane proteins, for example. So labeling and modification is also on the scope of the company, and high throughput solutions in protein production, which means on-chip automation, but also robotic systems for the cell-free production of membrane proteins. Now, you're all familiar with the overexpression of membrane proteins and the synthesis of these difficult to express proteins in classical in vivo systems, of course, the prokaryotic system for the high yield production, the eukaryotic systems, but also yeast-based system for the production in and the subsequent integration into membrane compartments. But the problem starts when producing toxic proteins, constantly active pore-forming proteins, and those proteins which are really difficult to express. So therefore, we decided to produce novel cell-free systems based on all of these in vivo systems. So we have the corresponding prokaryotic system, we have a corresponding cell-free system based on Pichia pastoris, so from yeast, and we have the corresponding system from the CHO cells, SF21 insect cells, K562 cells, and HEX cells. So whatever cell-free system you need, you can purchase the system also from the company here. Now, the second advantage of the cell-free system is that they are devoid of any batch-to-batch variations. So we produce from one batch where we grow the cells in suspension, one batch of cell-free system which can be stored for more than 10 years. So for the next 10 years, you have the same basis for the generation of your data. And we add additives for energy regeneration, so you end up with higher yield of protein which is produced. So those are the robots in our company. We have the 96 well plates. We produce 96 different membrane proteins. Incubation oven is over there. And in cell-free system, the time course of membrane protein production is dramatically faster than in any in vivo system. So after 60 minutes, the maximum yield of the protein is produced. System can be scaled up to the 100-liter range. In the 100-liter range, you're working with kind of a wave back. So those are the high throughput and highly parallel system. And I think for single channel analysis, you don't need tons of proteins, but you need active protein. So for the starting of the synthesis reaction, you do not need to clone anything. So the entire procedure is free of any genetically modified organism. You can handle the whole cell-free system in technical surroundings and technical environments. As here, you don't need biosafety S1, S2 labs or something like that. You can even start with a linear PCR product. PCR product can be spiked into the cell-free system. Of course, there are not only the ribosomes, but also initiation, elongation factors, tRNAs, and so forth in the cell-free system, which we call lysate. And in parallel, which is a really user-friendly, convenient methodology, there's transcription and translation in the same one-pot reaction. Now, if you need the highest yields, we have the dialysis systems. In the dialysis systems, byproducts are constantly removed, and thereby the yield of the protein is dramatically increased. Just to give a figure about that, it's 1,000 to 1,500 micrograms per milliliter of membrane protein, which is produced in these systems. Unfortunately, the protein doesn't jump out of the reaction, so we still have to optimize that. You have to clean and purify your membrane protein prior to this subsequent downstream use. Now, with the Fraunhofer project, we did some miniaturization and on-chip protein synthesis. So we designed this type of chip where all of the ingredients from the nucleus were transferred to this transcription compartment. Messenger RNA is transcribed, polyadenylated messenger RNA is transported via a magnetic device into this washing station. RNA is washed and is further moved into this translation compartment by the magnetic movement. And in the translation compartment, the protein of interest is produced. So this is all in a sub-microliter range and on-chip synthesis of a protein. The chip can be applied to certain devices. And after having produced these proteins, we use them for spotting on chip surfaces to analyze autoimmune diseases where the sera from the patients address membrane proteins. This is a project with the Charité. Now, this brings me to the next interesting aspect in cell-free system. Our partner here is the company Exazyme in Berlin, also a startup in the field of artificial intelligence. So how can we speed up the design of novel enzymes, for example? And nature has not invented the living cell to produce highly active enzymes. It's rather the opposite way around. The highly active enzyme kills the eukaryotic cell efficiently. So the more enzymes we design by artificial intelligence, the better we kill the living cell. So this is a classical approach for a cell-free system. We design the protein of interest, pore-forming protein or enzyme. We synthesize the DNA completely in a chemical synthesis, and then we spike in the DNA. We produce the protein after functional analysis. In an iterative way, artificial intelligence again designs the next round of proteins, and by expression PCR, we have the linear templates. We produce them in the cell-free system, and so forth. So this is an interesting evolution of membrane proteins and tailor-made enzymes. So we have some partners in the Nordic countries in the field of enzyme design. Now, you are interested in active membrane proteins that can be produced in the prokaryotic system. This is our approach with the prokaryotic system. First of all, we have different strains of prokaryotes. Each individual prokaryotic strain does the overexpression of an individual chaperone. So we separate GOEL, GOES, DNA K, DNA J, and so forth. So we produce 5 different lysates in parallel. We mix these 5 lysates, and as a result, we have all of the chaperones in the prokaryotic system. On top of that, we do further optimization steps. I have to apologize for the German over here. So we can shift the pH, we can shift the ion concentrations, we shift the dilution of the material, the lysate itself. If you dilute the lysate, there's a slower onset of protein production, better folding of the protein, and we can produce the protein at various temperatures. Lower temperature is better for folding of certain difficult to express proteins. In the end, we combine everything, and now you know why we need these robots here. 24/7, they produce these proteins in prokaryotic cell-free systems, and on top of the maximum yield of function protein, you can then add the nanodiscs for embedding of the membrane protein into a membrane. Compatible solutants does not only mean chaperones and detergents, but it also means very mild detergents like Bridge, DDM, and other substances as sugar molecules, polyalcohols, and so forth, which are beneficial for improving the functionality and solubility of a membrane protein. Now, with the eukaryotic cell-free systems, which are produced in-house, the thing is completely different. Of course, we do not add any detergents. We have vesicles derived from the endoplasmatic reticulum, so there's an active translocon, and all different types and subtypes of membrane proteins, from multimeric membrane proteins. You can produce them in parallel as the translocon translocates those proteins into the vesicles, and this is clearly depicted here. Upon addition of the signal sequence, you address these ER-derived vesicles that are already present in the eukaryotic cell-free system. So in this way, there is a chance to produce different membrane proteins, embed them in ER-derived vesicles, and analyze them by their function, like TRPV1. And the bleaching experiment clearly shows it's not a passive integration, but rather an active translocation of the protein of interest into these vesicles. Some years ago, we published these results with the excellent work of Srusha and Dondar Party here. We produced a KCSA as a tetramer already in the cell-free system, completely assembled, functionally active. And by spiking in 14C leucine for labeling of the tetramer, we could clearly show the cell-free synthesis of the tetramer in these vesicles. And this was followed by a subsequent functional analysis at that time with this Orbit 16 machine. So painting of the bilayer, and those are the vesicles from the cell-free system decorated with the multimeric protein of interest. By a direct fusion of these vesicles, you could analyze the functionality of this membrane protein. So the overall procedure takes 2 hours. 1 hour for the protein production, 1 hour for the analysis of the data. This is another example. This is the thermostable direct hemolysin. So we focus on these hemolytically active proteins, really difficult to express protein, tetrameric protein. And by adding 14C labeled leucine to the cell reaction, you can directly monitor the monomers, the dimers, but also the tetramers in the system, and vesicles are then used for subsequent functional characterization of this membrane protein. Now, yesterday we heard excellent talks on the G protein coupled receptor. So this is the upcoming topic now for us, and I'm a very long time in the field of G protein coupled receptors, in particular olfactory receptors. Here we produce the G protein coupled receptors in a cell-free system. We integrate the GPCRs in these lipid vesicles from the ER. And we use them either for ligand detection, but also for fluorescently labeling for the generation of biosensors, and simultaneously read out of optical and electrical activities of these GPCRs produced in cell-free systems. Now, as one example, this is the endothelin B receptor. We simply fused the GFP to the very C-terminal part of the receptor, was produced in the cell-free system. You can see the immobilized vesicles on the chip surface decorated with the G protein coupled receptor, which was produced on chip in this case. Now, how can we analyze ligand binding? And this is another option, not an impedimetric measurement, but is rather an optical readout of these immobilized G protein coupled receptors. First of all, we produce the receptor in the cell-free system, which is fused to GFP, shown here. We add the ligand, which is endothelin 1, which is labeled by Psi3, so it's second wavelength, and we do not see any binding of the endothelin 1 to the receptor, which is due to the fact that the ligand binding site is inside of the lumen of these vesicles. So we're working with ER-derived vesicles. There should be no ligand binding once you apply the ligand from the outer side to these vesicles. So we had to establish a methodology for the very mild treatment of these vesicles, for perforation of these vesicles. So it's a kind of a detergent-induced ligand binding to a GPCR. So we add a low concentration of bridge 35.03%, which does some perforation of the vesicles, but does not destroy the vesicles. And you see then the induced ligand binding, which is the red color over here, from the endothelin to its cell-free produced G protein coupled receptor. Now, we don't want to deal with these GFPs or YFPs and so forth. We want to have more defined labels in the membrane protein at defined positions. So in principle, there are 2 ways of labeling of these proteins in the cell-free system. First of all, you could simply add the pre-charged tRNA, and the fluorophore will then be introduced statistically into the protein of interest. There are a few examples where this is the method of choice. For example, the ribosome itself accepts perfectly this rather bulky fluorescent moiety, so body pile can be embedded in any membrane protein in this eukaryotic cell-free system. There's absolutely no problem. Also, the biocytin and the biotin moiety is quite well accepted by the ribosome, although it's a bulky substrate too, but the ribosome accepts that, introduces that into the protein of interest, and you can then use the streptavidin peroxidase, for example, for antibody-free detection of a membrane protein. which is of particular interest for these viral membrane proteins where there was no antibody available, but we should demonstrate the expression of the protein, and this was the method of choice. You see fluorescent is, I would say, in the same sensitivity range in comparison to autoradiography, sometimes even better. This is the body pre-charged tRNA, which was added to the cell-free system. You might also get rid of this band if you do an RNase treatment prior to loading of the protein shells. Now, a second way of introduction of fluorophores into self-free produced membrane proteins is the site-specific incorporation. This is the method of choice for our systems. We prefer this one. Here we design a tRNA, we exchange the anticodon against the AMBER anticodon, we spike in the pre-charged tRNA, in the end the label is in one defined position in your membrane protein. We further enhanced this system by adding these kind of enzymes, the amino-acyl tRNA synthetases. So they pre-charge only their substrate, which is this specific tRNA, and this way the tRNA is recycled and always pre-charged with a fluorophore, which is then site-specifically incorporated into your membrane protein. Now, nature has invented 3 stop codons to support us scientists. This is a very lucky situation. We only need these 2 stop codons for termination, and we use the AMBER stop codon for reprogramming. So by expression PCR, we add the AMBER stop codon at a defined position in the membrane protein. We then use the system for the integration of this non-canonical amino acid, and we use para-aceto-L-phenylalanine. So this opens a broad range of further downstream options to spike in these rather bulky substrates into your membrane protein by simple click chemistry. And as shown here, you have more than 90% incorporated in the protein, and click chemistry shows nicely the addition of these bulky chemical moieties. So the next step of enhancement was this bioorthogonal system. We're working with a eukaryotic background, so we have the insect cells or the CHO cells, and the insect or CHO endogenous synthetases, they pre-charge the endogenous tRNA with 20 canonical amino acids. We add on top of that a prokaryotic amino acid tRNA synthetase and a prokaryotic tRNA. So in this way, we have a fully bioorthogonal system, and there's only pre-charging of the prokaryotic tRNA with the non-canonical amino acid, which is, in our case, para-aceto-L-phenylalanine. So with this system, this doesn't cost anything because you overexpress a completely useless prokaryotic enzyme in a eukaryotic cell for the preparation and disruption of the eukaryotic cell. And then afterwards, the prokaryotic enzyme shows its maximum activity of pre-charging the prokaryotic tRNA. It's an interesting patent, useless enzyme in a eukaryotic cell. But in this case, after lysate preparation, it does the pre-charging introduction of the para-aceto-L-phenylalanine, which gives you these further downstream options for chemical reactions. We prefer the strain-promoted addition of chemical components, fluorophores and so forth. Copper is not really beneficial for biological systems. So we implemented this E. coli tyrosyl tRNA synthetase, and I will briefly go through this. We added some mutations to have open access for those substances. So para-acetyl-phenylalanine is one option for labeling, but also propargyloxy-L-phenylalanine can be introduced in the protein. So you can go in 2 directions with the click chemistry downstream. And there are further options, of course, if you want to introduce photoreactive groups, side chains, redox-activated side chains, metal chelators, or these hydrophobic side chains. I already mentioned these guys over here, biocytine and body pie labels. Those modifications are mainly based on the tyrosine or on the phenylalanine, so are lots of options for site-specifically introduction of these amino acids into your protein of interest. So here's another example with the G protein coupled receptors. In this case, we added the nanoluciferase to the very C-terminal part of the protein. We introduced the non-canonical amino acid at this particular position, which is the body pie. And in a bread assay, we added a substrate for the nanoluciferase. Upon ligand binding, we see a change in the conformation of the GPCR, and the readout clearly shows that there was activation of a GPCR detected by this bread methodology. This is a very interesting method to be combined with other methods on a chip surface, impedimetric assays, for example. Here I have to mention we need open access for the substrate to the nanoluciferase, which is possible in these vesicle-based systems. Now, if you don't need any label in the membrane protein, feel free to shift your label to an antibody which is designed against the membrane protein. And this is in the scope of our research in using those membrane proteins for cancer treatment. So we use the membrane proteins as typical tumor markers. We produce different formats of antibodies in the cell-free system. Of course, we do not sacrifice hamsters, I have to mention that, but we use the CHO cells and we prepare the cell-free systems from the CHO cells, and we apply then the production of antibodies to this system. Here is the example of how to attach a payload to the antibody. Of course, we again introduce the para-acetyl-L-phenylalanine, and for the detection of the antibody, we use the dilite 650. So feel free to couple whatever label you want to couple to the para-acetyl-phenylalanine. You can see the fluorescently labeled antibodies and different antibody formats. In a second step, in a theranostic approach, we exchange the fluorophore for the cytotoxic payload in the same antibodies. Those antibodies produced in a cell-free system can be analyzed. So we spin down these vesicles harboring the cell-free produced antibodies in the luminal part. We exchange the buffer, we release these antibodies from the luminal part of the vesicles. We apply them directly without any further purification step to SPR analysis where membrane proteins are immobilized on the surface. And this shows nicely the binding of the self-reproduced labeled antibody to the membrane proteins. So this is the main focus of the company, designing antibodies against membrane proteins, use those antibodies with different linker combinations and produce these antibody payload conjugations with different conjugation strategies. We do not need the tagging and purification as we're working with the eukaryotic cell-free system. Those no endotoxins present in the system. We can directly apply then these antibodies to the cell lines harboring the membrane proteins or even primary tumors, which we get from the Charité, and analyze the effect of novel types of linkers, novel types of payloads, and different sequences of antibodies produced in high throughput. The overall concept of the company is, on the one hand, these antibody-drug conjugates, which I already mentioned, the tailor-made enzymes by artificial intelligence-based design of these enzymes. We produce membrane proteins for the assay development. The company is a member of the Diagnostic Net Berlin-Brandenburg, so we offer these membrane protein production service for all different types of downstream chip-based applications. The opposite way around is the analysis of messenger RNA-based vaccines. So we get the messenger RNA-based vaccines from different companies in Germany. I'm not allowed to mention the names, but you know them. We do the quality control for these messenger RNA-based vaccines. And this is now brand new in the field of the company, which will go in holding in another company, the membrane protein production service, a kind of an Amazon for proteins. Simply send me an email with your membrane protein sequence. Where do you want to have which label, which linker? We produce it and we deliver it within 7 working days based on these cell-free systems. Now we're also in the field of in vivo protein production, and I'm very happy that this year we had this common publication, thanks to Maria, Andre, Sushan, Niels, and the experts, and our PhD student Alexander, and this master student Maximilian. We were luckily expressing the human PEP-T1 followed by SSN-based electrophysiology. So whenever you need a cell line overexpressing a transporter, feel free to contact us. We will produce this cell line and deliver the cell. Now the shareholders are in the field of chemistry, so we can design whatever linker you need, whatever fluorophore you need, or several fluorophores combined in one position in a protein. Haixang Zhang is French. He lives in Strasbourg and produces these linkers. The expert for linkerology is Thomas Bruckdorfer from the company Iris Biotech. This guy you might know. Now, this is Sven Barke, expert in the field of antibodies, former glycotope employer. And in the clinics, we have Wolfgang Sauerwein for the clinical trials of antibody-drug conjugates, antibodies designed against membrane proteins. So we cover the entire pipeline here. Of course, we are not perfectly gendered, so we will only hire female scientists within the next year. Those are the four guys, the four founders of the company. So four guys, four beer in a pub in Berlin. So this is the name B4 PharmaTech. Thank you very much. You also find us here in the internet, and we would be happy either on the academic part of the research, but also to support your research from the company. Thank you very much.
Thank you very much, Stefan, for this really nice overview about cell-free synthesis and all these options and tools you can provide these days. Are there any questions? Was all.
So for cell-free synthesis of membrane proteins, do you need to add some membranes to the extracts, liposomes or something like that?
There is an option to add whatever you want. As you're working with an open system, you can spike in whatever lipid components you want. In the prokaryotic system, we prefer, first of all, this, as I mentioned, matrix for increasing the solubility, chaperones, and then add on top the nanodiscs. And you can play around with different nanodiscs. It's better to do it this way around than to start the other way around. If you simply add the nanodisc to a whatever system, you end up in misfolded non-active proteins. This way we show activity, and then you add the nanodiscs for embedding of the protein. It's the better way. In the eukaryotic system, we have these vesicles and active incorporation of multimeric proteins, 100% in one orientation because we use the translocon. It's not a passive embedment. You can even spin them out and exchange them or reuse them several times in a kind of step-by-step production of several subunits of a given membrane protein complex.
All right, thank you so much. Any further questions there?
Thank you so much for this very interesting talk. I have a question regarding the ADCs, which are obviously a very hot topic these days. So what would you say is a typical customer for these ADCs? Or what I actually want to know is what kind of like functional readout are people using to elaborate on the functionality of these ADCs?
The typical readout is the killing curve, and here I would really collaborate with you and your excellent machine for showing killing curves on chip surfaces, because here we have the optical readout so far, but by impedimic measurement, this is the brilliant readout for the antibody-drug conjugates, in particular as we are addressing the membrane proteins that are overexpressed in these cell lines, membrane GPCRs. I can name, say, 20 GPCRs involved in tumors. So these antibodies are designed against these GPCRs. We analyze the toxic components over here, which are then clicked via novel linkers, and it's more or less a field where you have to analyze lots of different linkers. Currently, we have 800 linkers in the library, and there's a different kind of, say, time programmed release of the linker. So the linker itself decides when will the payload be released in the internal compartment of the living cell. This is a hot topic, of course, you're right. And currently we analyze this with optical readouts here. We embed, for example, primary tumors in soft agar surfaces, and in the soft agar surface we also analyze the antibody-drug conjugates.
Hey, thank you so much. So, it's another question.
Thanks for the nice talk. I have a question regarding the microsome expression in eukaryotic system. So in our plant cell-based lysate, when we use malitin signal peptides for the soluble protein expression, we always detect the considerable protein present in the cytosol part, and we still don't have a conclusion. So maybe someone believes it comes from just not the vesicle not fully precipitated, and someone believes maybe the protein leak from the microsome. And I have feeling some protein just, even with signal peptide, is still darkly expressed in the cytosol. So I want to ask, have you ever detected such phenomenon in CHO cell, insect cell lysate? And do you have any comment to this?
Yeah, I have several comments on that. We have to take into consideration three different aspects. First of all, the medicine signal sequence itself improves solubility of a protein. So sometimes without any vesicle, you end up with a high concentration of soluble protein upon addition of the melatonin signal sequence, which can also be added to GPCRs, for example. On the other hand, if you have a high concentration of the vesicles in the system, you end up with the protein in the vesicles, but we have to also to keep in mind that the signal recognition particle should also be present in the system in an active manner. And this is recycled. And the lowest concentration is the signal recognition particle. So it's not a problem of the vesicles, it's more a problem of having a high yield of signal recognition particle that binds to the melatonin signal and transports the membrane protein into the vesicle. This is the second aspect. And the third aspect, you do not only end up with these vesicles, which have a typical diameter of 1 to 10 micrometers, but you also have some small-sized vesicles which cannot be centrifuged. And then the membrane protein is perfectly embedded, but the vesicle is too small to be precipitated. It's in solution.